Greetings everyone, my name is Saibal Ray and I am the current biology faculty from Nucleon Coaching Institute, and today we will be continuing with the previously left off video regarding your chapter 4, that is Principles of Inheritance and Variation. From the previous video, I hope that you have basically understood or grasped a bit of concept regarding your gene and genetics. Okay, but whom do we credit it for, or whom to give the credit for this massive discovery that gave us the opportunity on understanding what is actually a gene? That credit will forever go to an Austrian monk, that is Gregor Johann Mendel. And in his experiments we found out that there has been always a pattern for inheritance. Now you may tell me that if Mendel was so much successful at the point that when he discovered this massive research or this massive finding, why was it not more provocatively accepted by the scientific community? Well, there are majorly two reasons for that. The first is that the prevalence or the much more importance was given to Charles Darwin's theory of evolution, and along with that utilizing your mathematics into biology was something which was unheard of before your genetics and before Mendel's experiments. Okay, combining all these with the prevalency and the not the prevalency actually, but in a sense that everyone was much more fixated on the aspect of evolution and Lamarckism or Darwinism more, rather than understanding what has been or what is the experiment from a very small place, that is Austria. Okay, very small place of Austria, that is Bern, which we will come to know about it also. Okay, so in his papers he basically gave you the translation of the characters. Okay, as we have discussed about characters and as we have discussed about what are these, okay, he basically gave you a reason on why we get characters to begin with. Okay, and why characters are basically inherited. That is why he is called as the father of genetics. Okay, now what is the age? That is, he was born in 1822 and he died in 1884. Was born to a poor family of Moravia in Austria. He completed his research and basically his education in the same monastery, okay, wherein he joined as a monk in the church and became the head of the monastery at Brunn, Austria. Okay, this is where right now there is an institute, and at this present moment, okay, that is called as Mendel's Institute. Okay, Mendel's Institute of Genetics, which is located in Brunn, okay, or Brunn. Okay, during his period, why was Mendel successful? Okay, why was Mendel successful? We will come to who rediscovered it. Okay, why was Mendel successful? Mendel was successful because of two major reasons. That is, first, he managed to take only those characters which have a predominant effect. Okay, a predominant effect means that they were very visible, they were very morphological, it would be easy to track it. Second, the plant that he chose, which was garden pea or pea plant, okay, was self-pollinated. That means it showed selfing. Okay, and the second thing is that it had all the right characters on and all the parts that are required for you to make a proper note of, or to make a proper recording of. Okay, and in that recordings itself he took 7 characters, which we will discuss in the later. Okay, or I can show it to you also. What are those 7 characters? If I'll give you a simple trick, okay, on how to remember this. Okay, I'll give you a simple trick on how to remember it. First, we will be discussing about what are these characters, okay, that Mendel took while experimenting in the pea plant. Okay, then we will take, these are the characters that we will be taking. Then we will take what is the expression of these characters, that means what is the dominant trait of that character. Okay, dominant trait of that character. And then what is the recessive trait of that character. Okay, something that is not readily visible, or something that is not readily seen. Visible would be a wrong term. Something that is not readily shown. Okay, that is what the definition of recessive I gave in the previous class, right? So the first thing that we see is plant height, the height of the pea plant. Okay, the dominant trait for this was shown by tall, because tall were found to be in more. Okay, and the recessive was dwarf. There were 7 characters that Mendel took, and we will be discussing all of these. Okay, after this has been discussed, if you see a plant, let me give you the trick that will help you to remember this. If you see a plant, what is the first thing that you notice? How tall is the plant, right? Plant height. Then what comes is that when you have seen the plant, what is the first thing that comes to your mind after seeing how tall it is? What kind of flowers does it have, right? If it does possess any flower at all. Okay, so that will be our, where is the flower? Okay, flower position. Okay, flower position, where is the flower exactly? That will be axial, which is a dominant part, and terminal, which is the recessive part. Okay, axial and terminal, no need to get confused, they simply represent where exactly will you find your flower position. Okay, then we come to third. Plant height, flower position is done. Now when you have seen the flower, what is the other thing that you will notice? That is, if it is a pea plant, it must contain pods, right? Where exactly is the pod, or if there are pods, what is the color of that pod? Okay, so we will be discussing first pod shape. Okay, pod shape, which is inflated and constricted. These are the two contrasting characters, or not characters, contrasting traits. Okay, then we will move on to, after we have seen the shape, we can discuss what is the color of that pod also, correct? Color of that pod also. So in here it will be green and yellow. My bad, I have written it in blue. Okay, this is constricted. Okay, we have discussed 4, the rest is 3. We will be discussing that also. Okay, let me rub it. Okay, Now we will discuss about the rest. Okay, that is 5. We have discussed pod, correct? We have discussed the pod, where what it is, right? But what we have not discussed and what we have missed out is that we have told where is the flower, but we have never mentioned what is the color of the flower. So we will be taking flower color as well. Correct? Flower color here will be dominant, can be either purple, okay, and the recessive can be white, okay, purple and white. Then we move on to the 6th one, that is what is the shape of my seeds. Why? Because when we open the pods, what is the thing that we are going to get? That is the seed itself, correct? Now you can tell me that once you see the seed, what are the 2 things that comes to your mind? First, what is the color of the seed, and second, what is the shape of the seed? So these are the 2 characters that he has taken and we are discussing that. Okay, what is that? That is seed, shape. If you want to discuss shape first, okay, what is the shape of this? In a dominant form, you will always find it to be round, okay, and in this case you will find it to be wrinkled. Oh, my bad. You will find it to be wrinkled, correct? Now the only thing that is left is what is the color of the seed. Color of the seed, which is, again, there is the difference, okay? In here your dominant is yellow and your recessive is green, okay, for the seed color. Now, how many of you have seen yellow colored seeds of peas, your garden peas? If you tell me that you have seen more yellow colored peas, then that means you are lying. Why? Because we don't see yellow colored seeds very often, and yet they are the dominant condition. Do you know why? Do you know why? Because as per definition, I have told you that for something to be called as dominant, it must show a larger population, or it must be shown in a larger population, right? That means the frequency of these expressing is much higher, and yet you do not find yellow colored seeds more often. Do you know why that is? Because of something called as selectivity. Why? Because we prefer it to be green. We prefer it to be green, and hence we need it to be green, and in that sense we avoid the yellow one, even though it is the more dominant one. If you keep on breeding, or if you keep on selecting something that which you want it will eventually give you the results which you want it, correct? This is called as plant breeding, a variety or a variation of this genetics itself. Okay, going a bit off topic, but it was a very good example to make you understand why, even though by definition a dominant trait must be the one that is expressed in a larger frequency than your recessive, we sometimes prefer the recessive itself. Okay, okay, then moving on, like I said, his Mendel's experiments were not recognized properly. Okay, Mendel's experiments were not recognized properly because of the prevalency of Darwinism and the abstractness of mathematics, or including mathematics into your biology. He did not get the recognition he deserved. He submitted his paper in 1865, okay, where he detailedly expressed all of the features of genetics, that is, all of the features of his findings, and yet it was not properly recognized. It was not properly lauded, and in that way, he never got to see his work being celebrated. But afterwards, in the early 1900s, there were three scientists who came up on Mendel's paper again, and in that paper they found answers which would unlock the entirety of genetics itself. That is, the contribution of rediscovery of Mendel's papers goes to Hugo de Verais, Franz Correns, and Eric Shermack. Okay, these are the three scientists that are credited with the rediscovery of Mendel's paper. Okay, moving on. This is the university, Mendel University in Brno. Okay, these are the characters which we just discussed. Okay, and these are the crosses that we are going to discuss. Okay, now in that sense, let me explain to you in a very brief way what is a monohybrid cross. What is a monohybrid cross? What do you think about having a monohybrid cross? A monohybrid cross is basically when we consider a single character. A single character is considered we call it a monohybrid cross. So let's take an example to make you better understand. Okay, if we have all the examples from Mendel's experiment itself, why not utilize them? Okay, let's suppose we take the flower color. Okay, we are taking the character flower color. Okay, we are taking the character flower color. In that sense, when we take a flower color, just in an arbitrary sense, we can give it, let's say, R. Okay, this flower color is controlled by R. R being the gene. Okay, R is what? R is the gene. R is the gene. Okay, and now eventually, of course, this gene will have two variants. That means this gene will possess two alleles, mostly two alleles. That is, a dominant allele, let's say represented by R dash. Okay, it is represented by R dash, and a recessive allele represented by small r, or let's not do small r. Okay, we can simply do capital R also. Okay, so that you don't get confused on why we take capital R to be a gene and why we don't take small r to be a gene. Okay, they both are a part of gene. Okay, the only difference is that they show variable expressions. That is why they are called as alleles. They are the alternative forms of gene. Okay, I hope there is no confusion in this. Moving on, since we have taken these two, there is something called as a condition. Okay, what is that condition? If you possess, or if a genotype, first I should explain what is a genotype as actually. Genotype is the inherent inherent property or a feature of an organism to be either homozygous, I'll come to what is homozygous as well, homozygous or heterozygous. Okay, whether it is a homozygous or whether it is a heterozygous. Now pay attention in this. When we talk about how homozygous, we simply mean that homo means similar, hetero means dissimilar. There should not be any confusion in this case at least. Okay, homo means similar and hetero means dissimilar. Okay, however, zygous, have you heard about the term zygous? It simply means zygotes. Okay, and where do we get zygotes? We get it after fusion. So this is possible only in the case of sexual reproduction, correct? Zygotes are simply such entities, okay, which are developed or formed by the fusion of two gametes, fusion of two different gametes, correct, formed by the fusion of two different gametes. So if they possess the same type, then we call them homozygous. If they possess a different type, then we call it heterozygous. For a very good example, let me show it to you. Okay, let me rub this out. Okay, for example, we are XY and the females are XX, correct? Have you ever thought on how to express this, or how is this expressed? XY or XX? XY is expressed as male and XX is expressed as female. Okay, XY is expressed as male and XX is expressed as female. In that sense itself, this is a heterozygous and this is a homozygous. Why? Because when it makes gametes, when it is going to create gametes, it is going to create an X and an X type of gamete. However, when this creates a gamete, it will be X and Y type of gametes. That means you will possess two types of sperms, that is X and Y type of sperms. Hence the reason you are a heterozygous and this is a homozygous. Okay. Now let us move back to what we were discussing, that is the monohybrid cross. We took, or we are expected to take R dash and R, correct? R dash represents the color, which let's assume is purple. Okay, because we took Mendel's experiment and we are discussing that this is purple. Okay, and this one will automatically then become white, correct? Capital R then represents white. So it needs to be in a genotypic form. That means it can be either this, correct, and this can be either this. These are both your homozygous parents. Okay, this will become, if we take, this becomes your female and this becomes your male. Can be reversed also. There is no such thing as this has to be a female and this has to be a male. Okay, we can reverse it also, but for the sake of argument we take it to be a female, and for the sake of argument we are taking this to be male. Okay, now once we cross it, like I said, all these are organisms which will create gametes. Okay, and during gamete formation they are going to create these gametes. Okay, okay, these are the two types of gametes that they can create. And as I have told, in homozygous situation we don't get a different type of gamete, they create the same type of gamete. Okay, now by law of segregation and independent assortments, okay, we will be getting a combination, that is each and every one gets a probability or a possibility of recombining or fusing with the other one. Okay, it gets a probability of getting fused to the other one, creating what we call hybrids or F1s. Now what are these F1s? These are nothing but filial progeny 1. Okay, filial progeny 1 or filial 1. Okay, like if we don't say progeny, okay, it will still be the same. Filial 1 simply means progeny, filial itself means progeny. Okay, what do they create is they create something like this. Now this, however, is called as a heterozygous. This is a heterozygous. That means it is going to create two different types of gamete when it goes into gamete formation. Okay, so what happens if we try to cross these two F1s? Okay, because from here, from the previous experiment or from the previous example, how many types we are going to get? We are going to get 4. Correct? 4 possibilities, and all of those 4 possibilities are going to be this, or if I write it in a better way, okay, this will become this. Correct? All the 4 possibilities will simply be R dash and R. Correct? That means from this way, okay, and then from this case, what is the expression that you are going to see? This is something that is happening inside cells, and inside cells we are not going to see, or we cannot see. So what is the thing that we can see? That this will be, can you guess it? It will be purple. All the flowers that we have will be of purple color. Why? Because we took purple to be our dominant one. Correct? And since dominant is, I have explained in the previous class as well, dominant is a condition. Dominance is a condition when it is present with some other allele. Only then a dominant case is established, and the best example for this is a heterozygous condition. Okay, in a heterozygous condition, a dominant allele is able to express its own expression rather than allowing the recessive to be expressed. That means it could have been white, but the dominant allele did not let the R gene or the R allele to be expressed, that is white color to be expressed. Okay, this has always been the case for your hybrids or F1s. F1s are always, this is something that you must remember, F1s are always heterozygous. Okay, F1s are always heterozygous. Remember, I'm not talking about dominancy. Why? Because we will come to those exceptions as well. They are not always dominant. Okay, F1s are always, however, heterozygous. Okay, now, like I said, what happens when we cross these two? What happens when we cross two F1s? We get something called as F2. Okay, remember, the filial progeny second or F2 will always be obtained by selfing. This process is called as selfing of F1s. These are F1s. When we cross them with each other, we will get F2, and when we cross F2 with each other, then we get F3. Okay, and so on and so forth. Okay, so what happens in this? They will also create gametes. What are those gametes? Here you will have R dash, and here you will have R. Similarly, you will have R and you will have R dash. Give me a moment. Give me a moment. Okay, thank you. So we will get R dash. Okay, and in this sense, we get F2, correct? This will pair up with this. This will pair up with this. This will again pair up with this, and this will also pair up with this. Okay, so you still get 4 possibilities. Okay, you still get 4 possibilities, but now there is a slight difference. Of all the 4 possibilities that you get, okay, of all the 4 possibilities that you get, let me explain it to you in a bit of mathematics. Okay, like Mendel used. If you consider this to be 100%, okay, what is the probability that you will get in this? That is 50%. 50%, correct? The gametes formed will always be 50-50. This is by virtue of, this is only possible through virtue of law of segregation. This is possible that we get 50-50 by law of segregation. Okay, they divide in 50-50 terms. Okay, now to make it a bit easier, okay, let us understand it in this way. You have R dash and R, you have R dash and R. By probability, you will get half of the sequence or half of the progenies to be like this and the other half to be like this, correct? Here also you will get half R dash and half R. Now if you make the crosses, okay, this is crossing with this, this will cross with this, and this will cross with this. You still get 4 possibilities, but all of them will not get 1/4. Okay, half and half, you get 1/4, 1/4 of R dash and R dash. Okay. Okay. Then we move on to the other half and half with this, that is 1/4 of your R dash and R, correct? Again from this, again 1/4 of R dash and R, and the second you get is half and half for this, that is 1/4 of R and R, right? This is the probability that you will get. Can we add these two? Can we add these two? Because they are basically the same. We do add these and we get half R dash R. Okay. We get half R dash R, and to simplify it, okay, to simplify it, this is the genotypic ratio. How do we get it? We got 1/4 of possibility for R and R, correct? We got 1/4 of possibility for R